Would you like to briefly introduce yourself?
Yes, thank you. My name is Pablo Sinues and I'm Professor of Biomedical Engineering at the University of Basel.
And what's your research about?
I am a mass spectrometrist, and what we are trying to do is to develop new technologies for non-invasive diagnosis.
So what you do is you analyze the metabolites in the air you breathe out, or?
Exactly. So we use sophisticated machinery called mass spectrometry to analyze the components of exhaled breath.
And what components are in the exhaled breath?
Well, exhaled breath is a rather complex mixture, and what you can find in exhaled breath are small molecules called metabolites.
And what are metabolites exactly?
Well, metabolites are the end products of our daily life. So when we eat, then we have to process this food in order to convert it into movement, for example, or having this interview. And it's just the downstream process of all these chemical transformations.
So what you eat, but also like in medicine and stuff like that?
Exactly. So basically anything that we intake, that we eat, that we drink, but as you say, it also includes medication, for example.
So it's harder to analyze, but do you also have an advantage when you measure the breath?
Yes, I mean, there are several advantages or complementary features of exhaled breath. One is obviously that it's highly non-invasive, and you can have access to exhaled breath continuously. Then another advantage, for example, is that it's always in contact with the respiratory system, for example, and this is a great window into the metabolism of the lungs, for example.
Mm-hmm. So it's just for metabolites of the lungs, or is it also possible to measure like stuff in the intestine?
Right. So as I mentioned at the beginning of the interview, it's a rather complex mixture, not as complex as, for example, blood. And this complexity comes because you are totally right. It's a mix between metabolites from the respiratory system itself, but also molecules or metabolites that come from the bloodstream and therefore that had been in contact or produced, for example, with gut microbiome.
Okay. Yeah. So that's possible to measure like short chain fatty acids as well in the breath.
That is totally correct. So short chain fatty acids are relatively volatile, and that's the reason why they can be exhaled in breath. And mass spectrometry is a very sensitive and very selective technique. That means that we are capable of differentiating these molecules from other molecules in this complex mixture.
Mm-hmm. And with mass spectrometry, you can also measure the molecules quantitatively, or just if it's there or no?
You can at least relatively quantify them. That's correct. Mm-hmm. It's not just a binary yes or no answer.
And what are the challenges of analyzing the breath?
Oh, there are multiple challenges. One of them, for example, as you just mentioned, is to be absolutely quantitative. That's difficult technically. Then there are other difficulties, like, for example, understanding exactly the identity of the molecules we are detecting. We detect signals, but giving it the exact compound identification, that is not trivial as well. Then there are issues like, for example, as I said, it's a gas sample, so the handling of a gas sample is more difficult than a liquid sample.
Mm-hmm. Do you want to maybe explain how the measurement works?
Yes, so we have two different variants. One would be what we call direct analysis. So we have a mass spectrometer here in the hospital, and some of our patients come directly into the machine. And all they need to do is to exhale or blow into a sampling tube. which is very easy to do. And that's it. I mean, there is no sample preparation, there is nothing involved. You just exhale into the machine and you have a sort of electronic signature of your metabolism.
Mm-hmm. How long does it take?
Just a few minutes, 10 minutes or so. We ask for a few replicates to ensure that everything is reproducible, but not more than that.
Mm-hmm.
The second variant would be when the patients cannot come to the machine. These are big machines. That's a drawback of this technique. So what we have developed is collection devices, or actually it's just a simple bag, which the patients, in our case children, because we are located in the children's hospital, they just inflate this sort of balloon, and then we transport the balloon to the machine. And then we just deflate this balloon and we look at the composition.
So it would also be possible to do a measurement from home. You blow into the balloon and send it to the hospital?
That is what we are working on right now, yes. That's exactly the idea. We want to scale this technology, and the only way to do that is by doing this kind of approach where the people just inflate the balloon, put it in the mailbox, and 48 hours later, 24 hours later, you have a result.
Okay. And at what metabolites exactly are you looking for in your research?
We are focusing mostly in metabolites associated with drugs. There are certain drugs that require what we call therapeutic drug monitoring. These are drugs, for example, anti-seizure medication or chemotherapy, some antibiotics. They require a very precise control because they are rather toxic. And so what we are trying to do is to look at these drugs and metabolites of these drugs to understand how the patients are metabolizing these drugs.
So you can adjust the dosage of the drugs, like if of antibiotics, so you don't give too much or too little?
Exactly, that's the overall idea. We would like to give a sort of real-time feedback or quick feedback to the clinicians so they understand if they need to increase the dose or decrease the dose or whether they are in the right range.
Okay, interesting. Yeah. And that's also possible with the blood, or is it like a completely new thing you can do with analyzing the breath?
Actually, they do it currently with blood levels, mm-hmm. and that's the standard practice. What we are trying to do is to do the same thing in breath. So potentially we could do it more frequently if it's needed. And what we want to do is to, in addition to this information of the blood levels, we would like to give additional information of all the metabolites which are modulated by these drugs. So we can actually tell the clinicians, okay, this is the level of the drug, and in addition, these are the levels of other molecules which are modulated by the drug, which is additional information to what they are currently doing in clinical practice.
Okay. But earlier you also said it's possible to do a diagnosis with this method?
Yes, that's a second branch of our research activities. For example, we are looking into quite heterogeneous diseases like asthma, for example. And there are some open questions from our colleagues in the pulmonology department. They would like to characterize better different groups of asthmatic patients. There are certain patients, for example, who are not responding properly to the medication. They still don't understand why is that, what is the reason behind. So what we are trying to do is to give this additional information so we can phenotype our patients mm-hmm. in a better way.
Do you know, is it like unique, your method with analyzing the breath with mass spectrometry, or are there other people in Switzerland or in Germany who do that?
There are other groups, of course, working in the same field. other groups here in Switzerland as well. For example, there is in the University Hospital of Zurich, there is another machine which is very similar. Mm-hmm. And we run parallel studies with them. There is people in Germany working as well with mass spectrometry, slightly different techniques. So yes, it's a relatively small community, but especially after COVID, I think it gained quite a lot of momentum.
Okay. Yeah. Are there like big disturbance signals? Like if you breathe out, there must be also analytes that are located not in the lung or in the air of the lungs, but also in the mouth. Mm-hmm. Is that a challenge?
Yes, it can be a challenge, certainly, but that's why mass spectrometry is important, because you can separate all this noise from the information that you want to look at. So they might be present, but if you have enough resolution, you are capable of detecting that. Mm-hmm.
What are the, like in what concentrations are the metabolites abundant in the air? Is it like quantitatively big amounts, or is it really little amounts?
Well, it's extremely small amounts. So, I mean, and the dynamic range can be high in the sense that there are molecules which are more abundant. But we know that those in the lowest level of abundance, they are in the so-called parts per trillion level. And I like the analogy that this is equivalent to one little drop diluted in 20 Olympic swimming pools. So extremely small.
Are there already any practical things you can do with this method, or is it just research at the moment?
Well, we already... so we created a startup company out of these research activities, and they are in the process of bringing this into clinical practice. This is not a simple task, because you need to go through a lot of regulatory activities, but there is already one solution for the case of anti-seizure medication, which is already approved and is currently being rolled out here in the hospital and also in the University Hospital in Zurich.
From which medication?
It's called valproic acid. It's an anti-seizure medication. For epilepsy.
For epilepsy, okay. Mm-hmm. And that's what you said earlier, like to detect how much is there in the body and how much you need?
Exactly, that's exactly the idea. It's telling you how much there is in the body. Then the decision of whether you need more or less lies exclusively on the clinician, on neurologist in this case. But yes, that's the idea. They get this information on the same day of the consultation. Okay. This is also important.
Yeah. What are your next steps for your research?
Well, the next steps is that now that we show that this concept works for this particular drug, we would like to extend it to other drugs which have the same problem. As I said before, for example, antibiotics or chemotherapy. These are drugs that require therapeutic drug monitoring, so we would like to build a full menu of these activities.
Okay. Is also the diagnostic part, is it as far, like, also as far as the thing with monitoring the drugs?
That's a little bit more far away in the horizon, because, as you said before, currently therapeutic drug monitoring is anyways done in blood. So what we are trying to do is to develop something that is similar, whereas a completely new diagnostic tool that requires more time.
Okay, yeah. Which medical conditions could particularly benefit from this method?
Well, anything that has to do with respiratory diseases, it's an obvious target to look at.
Okay. And does the breath analysis also have something to do with personalized medicine?
Yes, it has to do a lot with that concept. For example, I explained you before the case of asthma, where under the umbrella of asthma, we know there are many different subgroups of patients. And that's exactly the idea, to try to characterize better these patients so we can provide the right medication and the right treatment to each of these different subgroups.
What about early disease detection? Do you think in the future it would be possible to detect diseases earlier with mass spectrometry?
Yes, this is a topic we are not working on, but a lot of people is especially focusing obviously in lung cancer, which is key, as you for sure you know. It's very important to diagnose lung cancer as early as possible before there is any sign of it. Mm-hmm. So there are groups working into this topic, and I could easily imagine that in the future there are screening methods for lung cancer using exhale breath.
That would be great.
Yes.
Can you maybe tell us a little bit more about how does mass spectrometry work, or how does analyzing the breath with mass spectrometry work in your setting?
Sure. I mean, if I want to give an example, I mean, in the end, mass spectrometry, what it's doing is weighing molecules. So if, say for example, that I have a list of people with their names and their weight, and then I invite any person, and then I wait, and then I look at my table and I say, okay, it's 53 kilos, then it must be this person. That's kind of the idea. By looking at the weight, you can know what the molecule is. And once you know what the molecule is, and as you say, you know the abundance in exhale breath, then you can understand all these biochemical processes that are ongoing in your body.
Okay. So it's like you measure the weight of metabolites in your breath.
That's exactly what we do, yes.
Okay. So thank you for your time and the interview, and goodbye and good luck for your research.
Thank you very much for your visit.